Good afternoon, everybody. Thank you for joining me today. I'm Professor Ben Hall from UCL, and I'm going to be about exploring cancer evolution with computational models. So, the purpose of today's session is an introduction to our new Computational Cancer MSc. This new MSc is inspired by the fact that cancer research is being transformed through the application of computational methods. This is having impacts across our understanding of cancer, from our understanding of the fundamental biology to the early detection, diagnosis, and treatments in the clinic. And the purpose of the new Computational Cancer MSc is to train you in the fundamental skills in computational and data science alongside cancer biology to support a wide range of future careers. Now, the structure of today's lecture is I'm going to start off by giving you a taster session, an introductory lecture intended to give you some insight into what it's going to be like to attend the course. And specifically, I'm going to be talking about pre-cancer evolution and aging tissues. The topics I'm going to be talking about is about how cancers evolve from healthy tissues and are shaped by the environments that they encounter as they're evolving, as they're progressing into tumors. I'm going to talk about evolution and competition and fitness of different mutations, and about how the fitness of particular mutations depends not only on the gene that is mutated, but also the stage of carcinogenesis and the immediate local environment in which that gene is being mutated in. And I'm going to talk about how this has an impact on sequencing interpretation and any treatment or diagnosis strategies that are built off the back of that. Once I've finished my introductory lecture, I'm going to go on to talk about the Computational Cancer MSc, give you an introduction, give you some information about that before opening the floor for questions. So, on to the lecture. And really, we're going to be talking today about cancer and the aging body. How we age is a really important question for cancer because aging is a risk factor for a great many cancers. Now, cancer is extremely common, with CRUK statistics suggesting that 1 in 2 of us in our lifetime will develop a cancer, or 1 of 2 of us will go on to die from those cancers. The relationship with aging, though, is more complicated because we are living in an aging society, as many developed countries are. So understanding how we age, how our bodies change, and what controls those changes is vital to understanding how cancers initiate. It's also vital to understanding patient samples because the aged body is not naive, it's not mutationally naive. And actually, that changes the way we would interpret anything we can collect from patients. The reason for this is that cancer data is inherently collected late. Cancers evolve from healthy cells. At one point in their lineage, the cells that grow into tumors are normal, unmutated, happy in their tissues. As we age, we pick up mutations. Those mutations can come from cell intrinsic processes, like the process of division that all cells undergo, or extrinsic processes like exposure to UV. Some of these mutations change the cell, causing them to evolve, causing them to gain new properties. And they can do this by multiple different mechanisms indicated by the graphs on the right. But as they're evolving, they change their local environment. And if they change their state, they change their growth behavior substantially, they change that environment and they change the selective pressures. Now, this evolutionary process will be going on throughout a person's life, but when we take a tumor biopsy or study the tumor, we're only looking at the end of that process. And the early stages of that process are completely unobserved to us. So we need to understand this evolution that occurs within the healthy tissue. Now, it's worth stressing that healthy tissues remain normal throughout life. What I mean by that is if I were to take a look at the tissue from an older person in their 70s or a younger person in their 20s, morphologically, the healthy tissue would look the same. You couldn't see the difference in a microscope. but those tissues are radically different in terms of the mutations they bear. Now, when you're young and your cells are unmutated, those cells are competing neutrally at homeostasis. What that means is that the cells can replace one another or be replaced by one another, but no cell has an intrinsic advantage over others. And they're at homeostasis because the tissues themselves are not growing or shrinking. They're maintaining their rough number of cells in the tissue over time. As we get older, mutations occur. As I noted before, they can occur from intrinsic or extrinsic processes. Some of these mutations are going to give cells an advantage. They're in that competitive environment. So that is to say, they will allow those cells to persist and expand by giving them enhanced ability to replace others or resist replacement themselves. Now, when we look at cancer risk, we know that cancer risk can vary between different groups of people, but also mutational profiles between people and groups of people can vary too, suggesting that there might be something in that mutational profile that will give us a hint as to what is driving those risks and what increases those risks. And this drives us to our key problem, our key question. What will we get from understanding how cancer starts in the healthy tissue? It gives us the opportunity to ask, can we predict if a cancer will develop in a person? Can we take a sample? Can we make a study of an individual and tell them their risk score? And if we were able to predict cancer risk, could we use this as an opportunity to intervene early? Could we use this to stratify groups of individuals who are at high risk so that we can label them within the clinical pathway? And so if they present symptoms which would be not worrying for people at low risk, and change our interpretation and change how we act on that information. Similarly, would it be possible if we knew what an early cancer looked like or a high cancer risk looked like to intervene early in terms of therapies? More generally, though, if we understood the evolutionary process that occurs before the tumors become clinically evident, are there opportunities in for therapy that are present, that are visible only in that unobserved process? That is to say, are there things we're missing when we look just at the tumor that we could exploit and develop new therapeutic approaches for? Now, we're in the cancer space, we're not working in a vacuum of theory. And one of the key heuristics or um, concepts within cancer biology are the hallmarks of cancer. Now, I've got the diagram with 6 hallmarks now. Actually, if you look at modern diagrams, they talk about 20 or more hallmarks, but these are key features that cancer cells uniquely have that define them as cancer cells. I've stuck with 6 because it's quite still a useful framework to look at, and it's easier to talk about 6 in the immediate thing, this context. They're still very meaningful because what we can look at them when we look at those hallmarks, those features of cancer cells that define them, they describe the properties of cells that make them cancerous. So that is, the cells sustain their proliferation. They can divide constantly. They have no limit on their division. They evade signals from the body that should suppress their growth, and they evade cell death or apoptosis. So they don't respond to their own aberrant growth appropriately. They additionally change their environment as they're growing. They can induce angiogenesis to support the growth of the tumor, and they can also create new features that allow for invasion and metastasis. This has been a very useful paradigm for thinking about cancers for a very extended period of time. But evolution is another powerful theory, and understanding how the two of them marry together is not trivial. Intuitively, you might think that bit mutations are inherently going to be cancer promoting because they promote the proliferation and the replicative immortality from the hallmarks. However, if all the mutations are is fit. If they don't have other cancer tumorigenic properties, those fit cells will compete in the tissue and compete with other cells that may even bear cancer-promoting mutations to replace them and protect the body. So actually, so some cells that are fit may not be promoting cancer, and they may actually be cancer suppressing. On the other hand, if the mutations do have tumorigenic potential, but are not fit, they're not going to be able to expand or persist in the tissue. And it's only the cells that bear mutations that allow them to be both fitter than their neighbors and promote tumorigenesis that are going to be able to progress into cancer. So these slightly orthogonal concepts are in tension. So that brings the question, then how can we start to study this? How can we start to think about this? And we're going to start by thinking about how cells grow in healthy tissues. We're focusing on squamous epithelial tissues like the skin, the esophagus. These tissues are maintained by a compartment of dividing cells in the basement layer. As the cells divide, some divide to give more dividing cells, some divide to give cells that differentiate. Those differentiating cells stratify, moving up through the layers, undergoing a differentiation process that ultimately leads to their shedding and loss from the tissue. Though the esophagus and the skin have different structures, with the skin with hair follicles and sweat glands, and the esophagus relatively flat, the processes that describe their growth and their maintenance are similar to one another. And a simple model for understanding this is the single progenitor model indicated at the bottom, where one population of dividing cells divides stochastically, either symmetrically or asymmetrically. When it divides symmetrically, a dividing cell can generate two more dividing cells, or generate two differentiating cells, or it can divide asymmetrically to give one of each. This model is homeostatic if the probability of producing two dividing cells and the probability of two differentiating cells are equal to one another. So as long as those two probabilities are balanced, we can actually maintain homeostasis with this population of cells. So how do we look at this in humans? How do we start to study the mutations of humans? Well, the answer is we can take biopsies of tissues from healthy individuals, either as a byproduct of surgery or from autopsy data from individuals who've died from other causes. We can take biopsies of these tissues, we can sequence those biopsies, and we can, if we collect the samples in the correct way, we can get a spatial picture of those biopsies with respect to one another. So we're able to take a section of tissue, say where the clones are, and see how the clones may be spread over multiple different biopsies. Now, a key thing to bear in mind here is that though this is very powerful, it's giving us a detailed picture of individual people, there are very few people typically in these studies. And that's a major limitation, considering, of course, these are people, they're not controlled in any way, and they have a variety of different lifestyles and features. The data sets these generate are sequencing data sets. Now, sequencing is a key tool in understanding human somatic evolution. And there are different techniques and different ways you can do this. You can take targeted panels. This means you pre-select a set of genes you're interested in, and you just look for variants in those genes. You can also look to whole exome sequencing or whole genome sequencing. Now, with each of these choices, there are trade-offs involved. One of them is whole genome sequencing tends to be more expensive than targeted panels. But also, targeted panels tend to be more sensitive. That is to say, you can identify smaller clones with a targeted panel than you can sometimes do with a whole genome sequencing. So there's a trade-off in the particular technique you do and the biases that can introduce. But regardless of which techniques you use, the outputs for this are a collection of variants, that is changes from the germline cell, changes from your initial state, and frequencies, that is the fraction of number of cells in the sample that carry those mutations. And so we're going to look in particular now at one study. Now, this is a comparison of skin samples taken from the UK and from people in Singapore. Now, this is interesting because the UK has a higher risk of keratinocyte cancers than Singapore, indicated by the graph on the left. Now, this study shows through a bioinformatics analysis that there's a higher mutational burden in the UK, but also a distinct mutational landscape. This is interesting for lots of different reasons, but this aligns with this observation of the higher keratinocyte incidence, keratinocyte cancer incidence. So what do we see when we look at this data? Well, the first thing we can see is the graph on the left is that the samples taken from donors in the UK have higher mutational burdens than those in Singapore. This is interesting and not intuitive. Singapore receives a higher amount of UV from the sun, and so we might expect the pattern to be the other way around, but that's not what we find. Additionally, in the signatures of the mutation are different. Now, signatures of mutation tell you what the process is that gives rise to those mutations. And so what we've got in purple and green bars here are different sources of mutation for the different individuals. And in the Singaporean samples, the mutational burden is dominated by what are known as aging signatures in purple. These aging signatures are cell intrinsic processes that we can't avoid. In contrast, if we look at the UK data, we see more of the green signatures, SBS7, which are associated with UV exposure and UV mutation. So the UK not only has a higher mutational burden, but we're also getting more mutations from UV exposure. We can, going beyond the global picture of mutation, we can also look at individual genes in this study. Now, the genes that are heavily mutated are common to both populations. What's interesting is that they're not equally common. They're not a mirror of one another. And in fact, we find that some genes are more common in the mutated in the UK and Singapore and vice versa. So you can see genes like Notch1, Notch2, P53, and FAT1, which all commonly mutated in the two different populations. But when we try to compare the frequencies of the two of them, on the right, you can see that the Notch1 and Notch2 mutations are much more common in Singapore than they are in the UK. And in the other direction, P53 mutations are more common in the UK than they are in Singapore. Finally, we can actually look at the shape of the mutation clones and the size of the mutation clones. Now, when we look at the Singapore data, we can see on the left, the clones are well spaced out from one another, and they are large in the tissue. So we see individual cells with mutations able to spread. This is actually consistent with simple models of mutational burden. If we start off with a small number of mutated cells, these cells divide happily without encountering one another for extended periods of time because of the lower mutation rate. And so they get to grow larger before they collide with one another. In the UK, where the mutation burden is higher, we see more small clones, tightly packed, taking up the tissue. And again, the simple models are able to suggest why this would be. If we start off with lots of mutations initially, when we simulate that out, we can see the clones grow large and interact with one another and be constrained by those interactions, those clonal collisions. So simple models are able to explain why we see some of the patterns we see in that data. But one of the limitations we have here is that we have few donors and many features. What I mean by that is we only have, in that case, about 10 to 20 different people in that study. But we have all of these genes that we've studied and all of these things that are going on in the different individuals' lives that make it non-controlled. This makes fitting in interpretation computational models from human data difficult. So to better understand the role of individual genes, we can look to model systems and experimental procedures. In particular, lineage tracing experiments induce an inheritable label in cells, which can then be used to track individual clones in animal experiments. I mentioned earlier the single progenitor model. Now, this is a very useful paradigm for understanding how tissues are maintained. In this model, the dividing cells in green can divide symmetrically to give two dividing cells or two differentiating cells. Differentiating cells go on to be lost, whereas dividing cells go on to divide, or asymmetrically to give one of each. The probabilities of symmetric division are balanced, so the tissue is homeostatic. Now, we can use this model in the context of what is known as a branching process and simulate out the behaviors. When we do this and when we do mathematical analysis on the model, we can see there are certain properties of the model that we can look for in the experimental data. So we expect to see a linear increase in an average surviving clone size over time. We see the loss of clones follow a power law. So we see a rapid loss of clones initially and followed by a slowing of that loss. And the overall number of cells that are labeled should remain constant over different time points. Now, these different features arise from specific properties in the model. So the linear rise in average clone size and the loss of survival of clones are related to one another. That is because we see a linear growth in surviving clone size because several clones are lost, and that gives us that together will give us the homeostatic behavior. When we look at the experimental data, we can confirm we see these features in the data. We can see a linear relationship in average basal clone size over time. We can see the number of clones per millimeter squared follows a power law going down, but the percentage of labeled basal cells remains constant over time, confirming that the model is reproducing key parameters. More than this, we can also look at the distribution of cells, clone sizes in the tissue, and compare that to simulations from the model. We find this single progenitor model is able to replicate those more fine-grained pieces of data as well. So not only replicate the global picture, but also be able to tell us how individual clones are growing. So we have confidence that this is a good model for understanding the healthy unmutated tissue. So how do mutations spread? And what does that mean for cancer? Well, I'm going to talk about two genes today. I'm going to talk about P53 and Notch1. Now, the interesting feature about both of these genes is that if you sequence the tumors of the esophagus, you find that both of these genes appear to be under positive selection. That is to say, the analysis of the tumors say these mutations are fit and implies that fitness is part of the carcinogenic process. P53 can be mutated in the tissue, and we can track the clones with similar techniques to the lineage tracing I just talked about. When we do track these clones, we find that we can make small modifications to a single progenitor model to bias it in favor of producing dividing cells. When we build a model in this way, we find that the model is capable of describing the observed data, both in terms of the global takeover of the mutated clones, but also in terms of the specific clone sizes, the number of cells we find number of clones we find of different sizes. So a simple modification to the single progenitor model where we suppress differentiation is sufficient to describe the P53 behavior. Notch also seems to spread through the same mechanisms. Again, the single progenitor model where differentiation is suppressed is capable of describing clone size distributions and global takeover. But we start to see something more interesting going on with Notch as well. So in experimental systems, we can see that Notch pathway mutations appear to be able to compete not only with other cells in the healthy tissue, but microlesions. So early forms of cancer, and is able to actually kick them out of the tissue. And the greater the coverage and takeover of these Notch pathway mutations, the greater the fraction of tumors that have been eliminated. Moreover, and more confusingly perhaps, Notch1 is actually selected against in the tumors. This can be understood through the single progenitor model and its properties. Notch1 has two effects on the cell. The first one we've seen already in the healthy tissue. It suppresses differentiation. This is a really strong fitness advantage for those cells that bear that mutation and enables clonal spread. It also reduces the rate of division. When it reduces the rate of division, this has a minimal effect in the healthy tissue. But those two properties, the change in division rate and the differentiation bias, have radically different effects in the tumor. In the tumor, the cells are already committed to being progenitors, so loss of differentiation further has no effect. But the division rate becomes much more important. And so mutations to Notch that can be extremely fit in the healthy tissue are actually selected against in the tumor. This transforms our picture of evolution and also our confidence in our interpretations of the tumor sample data. Because I remind you that the apparent selection suggested that both not P53 and Notch1 mutations were under positive selection and promoted the tumor growth. But when we break down the data into looking at the healthy and the tumor separately, whilst we find that P53 is under positive selection in both the healthy and the tumor, the fitness advantage of Notch1 in the healthy tissue masks the fact that in the tumor itself, the same mutations are in a negative selection. And what we see when we sequence the tumor is an artifact induced by the fact that we've effectively compressed time, and that Notch1 mutations are extremely common. So to conclude this lecture, as we get older, we accrue mutations through both intrinsic and extrinsic processes. Both the quantity of mutation and selection determine the clonal takeover, and we can understand the clonal takeover and dynamics through computational models. And the selection of a single gene is conditional on the specific environment and the mechanism of growth. So that concludes my taster lecture for the course. I'm now going to talk a little bit about the Computational Cancer MSc itself. So this course has been inspired by a data revolution that's taking place in cancer. And really, one of the key motivations here is how can we take the multimodal, high-dimensional patient data that is available now and is becoming available and will be more available over the next 5-10 years to enable clinical improvements. Within this, there are a number of different questions we need to address. How can we enable better patient outcomes from this data? How can we integrate this data into clinical workflows so it becomes a standard practice to collect data and analyze it? And finally, a more general question, how do we work with new data types that become available? That is to say, not only do we think about the data types that we can routinely collect today, but when new data types become available, how can we integrate them into our workflows? How can we integrate them to improve better patient outcomes? At the heart of all of these questions is the use of computational modeling and data science. The Computational Cancer MSc aims to teach cancer through the lens of computational biology, presenting the roots and the role of mathematical analysis in tackling this complex disease so that you can be part of that data revolution and you can provide the tools to answer those questions. We're going to be looking at computational science for early detection, diagnosis, and treatment. We're going to look at cancer development from age tissue to metastasis, and we're going to look at it all from the perspective of modeling and data analysis. The core modules will be covering data science, AI, and mathematical modeling, whilst you'll be able to do options in computational pathology, AI for medical imaging, and radiotherapy. This course has been designed so it is suitable for a broad range of different people with a broad range of different backgrounds, including clinicians who wish to learn new computational mathematical skills to assist their work, and interdisciplinary scientists. Whether you've got a life sciences background or a computational or mathematical sciences background, there is going to be important learning that you can do in this course, and this course will be suitable for you. All graduates will leave with skills relevant to research roles in academia and industry. The breakdown of what this course program is going to look like is in the first term, we will teach about cancer biology and therapeutics, whilst giving you the fundamental skills in working with cancer data and cancer modeling. In term 2, you'll pick different options ranging from digital pathology, AI medical imaging, radiotherapy, physics, and precision cancer medicine. You can pick and choose what suits both your interests, but also your specific background. Finally, in term three, we'll focus on a research project where you will work with some of the top labs at UCL on a new piece of research. UCL is very much the place to be studying computational cancer. UCL leads the UK in citations in oncology and is one of the leaders in computational biology and maths. Moreover, all the work we do in this area fits within a strategy of UCL where we look to engineer better outcomes in cancer by embracing engineering and computation to advance detection, diagnosis, treatment, and management of cancers. The work in the MSc is going to be supported by existing bodies like the UCL Computational Cancer Collaboratorium, which provides a hub for different activities that are taking place relevant to computational cancer. It has a regular seminar series, meeting monthly, where you'll see speakers from the UCL and the rest of the UK, leaders in the field, talking about their research in the area. There's an early career researcher support network and careers events. In fact, we just had one on Tuesday where we had speakers from AstraZeneca and Johnson & Johnson talking about their career paths and how they ended up in their position and the different advantages of those jobs, the advantages that computational science has given them. Finally, we have an annual hackathon where people can join and work on group projects together. I mentioned careers events, and obviously career paths and career development is extremely important. Now, the MSc in computational cancer will support a diverse range of different future careers. These include data and computational science roles in research and industry. I already mentioned the pharmaceutical companies who came to our careers event earlier in the week. They also support work in computational pathology and analysis within their clinical environment, as well as healthcare and biotech. Finally, the skills and knowledge you'll develop in this course will help you with a wide range of adjacent careers, including roles in things like venture capital, where people are looking for people with life sciences and computational science background to identify the best opportunities, for example. And I'd like all of you who are interested to apply online. You can follow the link given in this QR code. And I will finish the session by opening the floor to questions. Thank you very much, everybody.
I'll just kick off with a question about, does the program have formal links with biotech or pharma, such as GSK, specific startups?
So we have conversations with people in AstraZeneca. We've had conversations with people with Johnson and Johnson. We have speakers and coming from lots of different companies. So we don't have formal links, but we've developed a network of different industry partners who are interested in the course and have come to give talks. And in fact, one of the things I would encourage people to do is, if they're interested in learning a bit more about the kind of work that can be done, go to our website for the Computational Cancer Collaboratorium, and I look at the seminar series, and in fact, you'll see speakers from academia and industry, and some of those potential, partners that we have, or relationships we have existing with different companies.
Thank you. So from one of our guests, is there an option to do the MSc part-time? How many days on campus does that equate to?
I beg your pardon, sorry?
Is there an option to do the MSc part-time?
Yes, there is an option to do it part-time. That is available.
And how many days on campus would that be?
That would depend on the term, but it'd be 2 or 3 days.
And for the optional modules, how competitive is the selection process? Are there caps on the most popular modules?
So the options we've listed are available to everybody on the course, and so we are not expecting caps on any of those. There may be these are part of the course, and they're available to everybody.
How can candidates from a computational versus clinical background choose the listed modules in your slide?
So I think you should choose them based on your background and your interest. So the ones that tend to be based in medical physics, or more computational, will be more suitable for somebody coming from a computational background because they'll go deeper into the math and into the computation. And so I'd recommend you, if you have a computational background, to be seriously considering those modules over the others. On the other side, if you're from a clinical background, or if you're from well, particularly if you're from a clinical background, I would strongly encourage you to take the pathology and digital pathology modules because they are going to be very attuned to what your interests are, and I think they're going to be a broader interest in life sciences on the course as well. So really, I think you should drive it by your knowledge, your experience, but also how deep you want to go, because some of the AI modules will get very mathematical and some of the more biological models will get very biological. So you're best to play to your particular strength and your interest and background.
And what types of research projects have students undertaken?
So this is a new course for this year. So we've not run any research projects, but we would expect research projects similar to the ones in fact I run in my lab for other things, which include understanding that aging process. So some making models of clonal spread and competition within tissues. You would expect network model analysis. You'd expect AI approaches to image analysis or similar genomics and data science type projects too. We'd also expect that those projects would probably be co-supervised by people both from a computational background, but also from an experimental background or clinical background, where they may be able to provide the data that you'll be working on.
Thank you. We'll just hang on another minute in case any more questions come through. I'm also just putting an email address in the chat for anybody who wants to follow up the lecture with any further questions or a message. Oh, sorry, one more. Do you have any examples of the kind of research projects we would do in term 3? Maybe.
So yeah, I just gave a handful of examples already, but I think that's it. So you'll be working with data sets that are provided by people in the Cancer Institute, um, and theoreticians based elsewhere. You'd be working on things like if you're working in my lab, you'd be working on evolutionary modeling, gene regulatory networks. In other labs, you might be working on AI or more data science type projects. It varies from lab to lab. Now, one thing I should stress is that this is very much a joint program between the Cancer Institute and medical physics, where Cancer Institute obviously has got a huge amount of work and analysis and data, where medical physics has the computational and mathematical expertise. So different domains being brought together to solve this key problem. Thank you everybody for attending. If you have any questions, I strongly encourage you to follow up with the email address that's been given in the chat. You can also find me on LinkedIn and feel free to reach out and contact me. Thank you very much, everybody.